All right, welcome back. This is going to be our last topic for quarter one. All right, so we're going to spend the rest of the week, including today, Wednesday, and then Thursday and Friday, looking at what's called chi-square analysis. And why we're doing this now is we just talked about different inheritance patterns and linked genes, and we looked at how we can use pedigrees to help establish what a specific inheritance pattern might be for a trait. But the big trade-off with that is that you need a whole big long history of whatever organism you're using to track the trait. You need a history that goes back in the past. And that gets to be kind of troublesome, um, especially if we're doing genetic experiments. So what this test is, and it's a statistical test that we'll do on data, so like information that you got as a result from an experiment that you did. And we're going to see how well the results of our experiment match our expected results. So for example, if we were doing a genetic cross between pea plants like Mendel did, and we um, were going to cross two individuals that we thought were heterozygous, and we got offspring. We could then look at the offspring and the frequencies, the ratios of phenotypes in the offspring, and compare that result to what we thought we should get based on like a Punnett square of the parents' genotypes crossed. And we could do a chi-square analysis, and the chi-square analysis would say to us, our results consistent with what we expected or not. So a chi-square test can be used in genetics, and we'll use it in other places in third quarter. So uh, you can predict genotypes based on probabilities. What are the expected results? The chi-square test is a form of statistical analysis that we use to compare the actual results, so like the pea plants that were actually raised from seedlings, what we observed, with what we expected from a Punnett square. One thing to note is that chi-square, that is a number, and it's represented by this symbol. Chi-square is the name of the whole variable. You'll never be taking the square root or solving for x. Okay? So a lot of times students get confused with that. Chi-square is the name of the whole variable. So we're never going to take the square root of it. That doesn't really have any meaning to us. So the idea is that if the expected results for like a genetic cross, as far as what the proportion of phenotypes are, and what we observe from actually doing the experiment are the same, then our chi-square number is going to be 0. So that's if our observations and our expectations perfectly align. So if the chi-square value is 0 after doing this test, or is very small, then our observed results are going to support our hypothesis, which is what we expected the results to be from our Punnett square. If the chi-square number is big, that means that our observations and our expected results from our Punnett square are incorrect. And so our Punnett square is probably incorrect. And that means that either the genotypes that we hypothesize for the parents are wrong, or potentially that this trait is being inherited in a different way than what we suggested. So by calculating the chi-square value, we are determining if there is a significant difference between expected and actual values. And so that's the key here. Chi-square tells us if there's a significant difference between what we expected to happen and what actually happened from observation. So, in calculating chi-square, the first step is going to be determine what you expect and what you observe. So, your observed or actual values will always be given to you because these are things that you could get from your data. There's no calculations involved in figuring out observed values. This is what is going to be given to you in every question. The expected values are based on probability. And so with genetics, this means Punnett square most of the time. So think observed values I get from data that's given to me explicitly. Expected values I will get from making a Punnett square. So here's an example. Let's say we're doing a lab about tongue rollers, and we observed that there was 90 tongue rollers in a group of 100, and then that there was 10 non-tongue rollers. So that's our observed data. For our expected data, suppose that the parents of these individuals were both heterozygous. So using a Punnett square, we would expect 75% of the total offspring to be tongue rollers and 25% to be non-rollers. So that means that we would expect 75 out of the 100 to be tongue rollers and 25 to be non-tongue rollers. Since I just took my expected frequencies of each and multiplied it by the total population size, 100. So here we have our observed 90 and 10, and our expected would be 70 and 25. That's all there is to it so far. So this is what it would look like if I put that information in a table. It's where we expected, this is what we observed. Now we're just going to plug these numbers into a calculation, and we're going to figure out the chi-square number. So we're going to use a specific formula to figure out the chi-square number, and it is on your AP Biology formula sheet. So I'll reshare that, and we'll use that and go over that in our Zoom call. But the formula is just that we are going to take the sum of all observed minus expected values squared divided by expected. We'll add all of those up. So for each different category, for example, tongue rollers or non-tongue rollers, for each category, we're going to take the observed in that category and subtract it from the expected in that category and square that difference and divide it by the expected of that category. And then when we do that for each category, in our examples, there's 2 categories, we'll add those 2 values up, and that's going to be our chi-square number. So here is what it looks like on your formula sheet. And they just use O and E instead of observed and expected. And the sigma, the summation notation, just means you're adding up that calculation for each of the categories. So here's an example using the same data from before. Okay, for tongue rollers, this is the first category. Observed was 90, expected was 75 squared, divided by 75 equals 3. And for non-rollers, observed was 10, expected was 25 minus squared, divided by 25 is 9. And then we add up the result from each category. 3 plus 9 is 12. So our chi-square value is 12. Now, just without looking at it qualitatively, 12 is big. So that means that our observations, our expectations are kind of far apart from each other. Okay, so now we have our chi-square number. But how do we interpret it? The last thing we need to do is interpret it, which doesn't really involve any other calculations, but it's making a interpretation of that number. Where is the threshold of that number being big or being quote unquote little? Well, that's what we're going to find out. And that involves determining the degrees of freedom. So the degrees of freedom for our experiments is always going to be the number of categories or the number of different possibilities minus 1. So for example, in this problem, there were 2 possibilities. Either there were a tongue roller or a non-tongue roller. 2 categories. So 2 minus 1 would have a degree of freedom of 1. All right, and so now we have our chi-square value of 12, and we have our degree of freedom number 1. And we'll go back to our AP Biology formula sheet to a little piece that looks like this, which is called the chi-square critical value table. And what we're going to do is we're going to go to the column with our appropriate degrees of freedom here, and we're going to compare our chi-square value to this number in the first row. Okay? So we call this number here a critical value. So this is where we're going to compare a number to. If our chi-square number is greater than the critical value, then that means there is a significant difference between our actual and expected values. And so whatever our hypothesis was is incorrect. If the chi-square number is less than the critical value, then there's not a significant difference between what we expected and what we observed, and our hypothesis is correct. So in our example, our chi-square number 12 is greater than that critical value. So there is a significant difference between the observed and expected populations. And that means that our hypothesis as to the genotypes of the mother and father, that they're both heterozygous, is probably incorrect. Okay, that's what this is telling us. So in this video, I just wanted to go over this. We're going to do an assignment here where we're going to practice calculating some chi-square numbers, and then tomorrow on Thursday, we're going to apply it to something that we looked at this week with linked genes and stuff like that.